We now move on to the gastrointestinal secretory products. The first two of which is gastric acid and intrinsic factor, both secreted by the parietal cells. The parietal cells are also known as oxyntic cells. They're present in the stomach and they mainly produce two critical substances, the hydrochloric acid and the intrinsic factor. The hydrochloric acid mainly converts pepsinogen into pepsin and this activates the protein digestion. So here are your parietal cells in the fundus and the body of the stomach, and they basically do four things. What the parietal cells do is they convert the pepsinogen into pepsin, which activates the protein digestion. It provides an acidic pH about 1 to 2 in the stomach, and it kills bacteria and pathogen, and also helps in iron absorptions because it converts the Fe3 plus into the Fe2 plus form. Right? Of the four things here, knowing the regulation of the parietal cells secreting this hydrochloric acid is also very important. So let's get right into that. So remember that for regulation of hydrochloric acid secreted by the parietal cells, we need these two things. It's increased by histamine H2 receptors, which is the strongest stimulator of its secretion, the hydrochloric acid, and it's also increased by the vagus nerve stimulation or the acetylcholine and gastrin. So gastrin, vagus nerve, acetylcholine, and histamine H2 receptors through the H2 receptors, these three things increase the secretion of gastric acid, and it's decreased by somatostatin, secretin, prostaglandins, and GIP. Now, histamine H2 receptors is the strongest stimulator, and the source is the enterochromaffin-like cells. So look at this here. Gastrin increases acid secretion primarily through its effects on the enterochromaffin-like cells, leading to histamine release, rather than through its direct effect on the parietal cells. So the source of this histamine is enterochromaffin like cells, and the mechanism is that the histamine causes the H2 receptor on the parietal cells to increase the CAMP, and that increases the hydrogen ion secretion. Why is this the strongest stimulator? Because it amplifies the effects of both acetylcholine and gastrin. So histamine amplifies the effect of acetylcholine and gastrin. So think of it as a final common amplifier. We can use H2 blockers like ranitidine and famotidine to decrease this acid. Now we also have something called as the vagus nerve, the acetylcholine, and the source of this vagus nerve or acetylcholine is basically parasympathetic innervation. So acetylcholine works on the M3 receptors on the parietal cells, which increases the calcium 2 plus secretion, and that leads to this calcium increase leads to the increase in acid. This also does two indirect things. The acetylcholine also works on the enterochromaffin-like cells to increase histamine, and the acetylcholine also works on the G cells to increase gastrin. So this acetylcholine also has a double effect on the gastrin and on the histamine through the enterochromaffin cells. So think of the vagus as a triple stimulation because the vagus is working on the acetylcholine, and the acetylcholine is itself working on the M3 receptors on the parietal cells, but it's also working on the enterochromaffin cells. It's also working on the G cells. So it's a triple stimulation. Then we have gastrin. This gastrin it comes from the G cells in the antrum of the stomach. If you can see here, these are the G cells in the antrum of the stomach, and this leads to our gastrin. So the G cells in the antrum of the stomach goes to a receptor called the CCKB receptor. Nothing very special, nothing to get confused about. Just look at this here. So we have something called the CCKB receptor. The gastrin works on the CCKB receptor. That increases the calcium and that increases the acid. And another indirect effect, but very important, to remember here that the gastrin also works on the enterochromaffin-like cells, and this also increases histamine, and this also increases a lot of acid. That's why gastrin is so powerful, and that's why in Zollinger-Ellison syndrome, the gastrin tumor basically leads to massive acid. You have the Zollinger-Ellison syndrome where you get a gastrin tumor, and that leads to massive acid. Now we talk about we've done with the increase. Now let us go through the decrease. Now, the gastric acid is inhibited by somatostatin. Now, somatostatin, we're talking about the somatostatin now. Somatostatin is produced by the D cells in the stomach. So look at these D cells. These D cells in the stomach produce somatostatin, and the somatostatin was working as a break, right? So whenever you have low pH, you know low pH means an acidic environment, the D cells secrete somatostatin. And what that somatostatin basically does is it does three things. This also works through triple inhibition. The somatostatin works on the gastrin cells, the G cells, and decreases them. So there's decreased gastrin. It also decreases the histamine from the enterochromaffin cells, and it also decreases direct acid secretion. So the somatostatin applies a brake on the gastrin. It works on the histamine. Puts a brake on that, and it also puts a brake on direct acid secretion. The next thing is prostaglandin. Let's also look at prostaglandins here, right? Before prostaglandins, let's work through the secretin. Now, secretin is produced by the S cells in the duodenum, right? Let's look at the S cells in the duodenum. So these are the S cells in the duodenum. Secretin is released from the S cells in the duodenum. The secretin is triggered by the acid entering the duodenum. So when acid enters the duodenum from here, this S cell is triggered to form the secretin, and the secretin basically leads to decreased gastric acid, right? It leads to decreased gastric acid and it increases pancreatic hydrogen carbonate. So this secretin, as you can see here, this secretin comes and it goes and increases the hydrogen carbonate coming from this pancreas and it decreases the gastric acid. So it basically neutralizes the acid that is coming from the stomach. The next thing we should look at is the prostaglandins that also create an inhibitory role. So they are decreasing the secretion of acid. Now, prostaglandins, what they do is they have a protective role. How do they play a protective role? The prostaglandins decrease the CAMP in parietal cells, and this decreases acid. The protective role comes from increased mucus, increased bicarbonate, and increased blood flow, right? So the prostaglandins do three things. They increase mucus, they increase bicarbonate, and they increase blood flow. Now, what you've seen that NSAIDs cause gastric ulcers. How do NSAIDs cause gastric ulcers? NSAIDs inhibit prostaglandins. They decrease this protective effect and they cause gastric ulcers. The last inhibitory role that we're going to study is GIP, the glucose-dependent insulinotropic peptide. The source of GIP is K cells in the duodenum and jejunum. Let's look for our K cells here. I'm not sure if they're present in this image. Oh, they are. They're just here. These K cells in the duodenum and jejunum secrete the GIP. So GIP is secreted by the K cells. And when is this triggered? Whenever you have fat and glucose in the intestine, the K cells are triggered to produce GIP. And what this GIP does is it decreases gastric acid secretion. Okay, it decreases gastric acid secretion. If food, especially fat, is already in the intestine, it slows the stomach down. Why? So the fat can readily be absorbed. Okay, it can be broken down and digestion can happen because fat needs some more time to be digested. So this is all the regulation of the parietal cells secreting the gastric acid. We now move on to the intrinsic factor. Intrinsic factor is also released by the parietal cells, right? Let me show you the parietal cells and the chief cells here. So this is a really nice image. I'm going to put it here and show it to you. So can you look at these parietal cells here and these chief cells here, right? So the parietal cells are secreting our intrinsic factor and parietal cells are secreting our gastric acid and intrinsic factor and the chief cells are secreting the pepsin, right? Here, these are the chief cells and these are the parietal cells. The parietal cells I told you are also known as the oxyntic cells. They're mostly present in the upper part of the gastric glands like the fundus and body of stomach. They're large, they're round, and they're a little pyramidal. They are strongly eosinophilic. Eosinophilic basically means pink due to abundant mitochondria. They have a central nucleus. You can see a central nucleus here. The unique feature is that they have deep membrane invaginations, which is basically intracellular canaliculi. And then we have the chief cells. Chief cells are basically present in the base of the gastric glands. They're found deeper than parietal cells. The smaller their columnar cells, they're basophilic, they're very bluish due to abundant ref endoplasmic reticulum. They have a basally located nucleus and they have enzymes called zymogen granules because they secrete pepsinogen, and this pepsinogen is converted to pepsin in the acidic environment. So when the parietal cells secrete their gastric acid or the hydrochloric acid, that converts this pepsinogen into pepsin, and also they secrete gastric lipase, but that's a minor role. They're blue because of the rough endoplasmic reticulum. So that's a little bit of histology about the chief cells and the parietal cells. We were talking about the intrinsic factor. So intrinsic factor is also being released by the parietal cells that are increasing, that are secreting the intrinsic factor. Now, the intrinsic factor basically what it allows is this intrinsic factor binds the vitamin B12. It allows its absorption in the terminal ileum. We can't see the terminal ileum here. And I told you that if there's loss of intrinsic factor, we get a vitamin B12 deficiency, which leads to pernicious anemia. And that leads to megaloblastic anemia and also wait, here, this is our picture of the pernicious anemia. So, loss of intrinsic factor leads to pernicious anemia, which leads to megaloblastic anemia and neurological deficits because of subacute combined degeneration syndrome. Okay, so remember that the parietal cells secrete the acid and the intrinsic factor. We go back here. We talked about chronic gastritis and pernicious anemia. We move on to our pepsin. Now, pepsin, as I showed you, the chief cells is secreted by the chief cells. Pepsin is secreted by the chief cells and it's converted, basically, the pepsinogen is converted into pepsin so that it activates protein digestion. Now, this pepsin basically comes from the chief cells. It helps in protein digestion. It's increased by vagal stimulation or acetylcholine, that's a local acid, and also by local acid. Pepsinogen is an inactive form. It's converted to pepsin active form in the presence of hydrogen ions. I think that's pretty self-explanatory. We move on to the bicarbonate. The bicarbonate is necessary to neutralize the acid. It's released from the mucosal cells of the stomach, duodenum, salivary glands, and pancreas, and the Brunner glands in the duodenum. Let me show you a picture of the Brunner glands in the duodenum because this is important. So look at this image here. The Brunner glands are located in the duodenum. They're found only in the duodenum. This is a hallmark feature. They're located in the submucosa, not the mucosa. So you can see that the mucosa is up here and the submucosa is here, and they're present in the submucosa. That's a very important exam point. If you see glands in submucosa, think duodenum immediately. The structure is that they have these compound tubuloalveolar glands and they're pale stained mucus, right? Their ducts open into the crypts of Libercan, which are basically the intestinal glands, and they're light foamy cytoplasm. As you can see, can you see this light foamy cytoplasm here? And they occupy large portions of the submucosa. Their function is very important. They protect the duodenum from the acid. They secrete alkaline mucus, which is rich in hydrogen carbonate, and this neutralizes the acidic chyme from the stomach. So, whenever you think of the duodenum, think about it as the acid shock zone, which needs protection. The Brunner glands are stimulated by the acidic chyme entering the duodenum, and they're also stimulated by the vagal stimulation. They're decreased when there is sympathetic activity. You can also have a peptic injury protection here. So whenever there's failure of these Brunner's gland, you can have duodenal mucosa damage, and that leads to a duodenal ulcer. Also, you can have hyperplasia of the Brunner's gland. They can form benign lesions, and you can have hyperplasia, and that may cause obstruction, but that's a rare point. But remember that whenever there's damage of the duodenal mucosa damage due to failure of these Brunner's gland, you get ulcers, mainly duodenal ulcer, obviously. So these Brunner's gland are very important. They neutralize the acid. They're increased by pancreatic and biliary secretion with secretin, and they're trapped in mucus that covers the gastric epithelium. So basically, the core concept is that the secretin is the bicarbonate hormone, right? So whenever acid enters the duodenum, the body responds to it by neutralizing it. And when the acidic chyme, the low pH acidic chyme enters the duodenum, the S cells secrete these S cells here. secrete the secretin, and this leads to targeting the pancreatic ductal cells here, and that leads to hydrogen carbonate and water secretion. And alkaline pancreatic juice flows into the duodenum, and also the action on biliary system. This basically cholangiocytes in the bile duct cells lead to increased bicarbonate-rich bile, so bile becomes more alkaline and watery. The net effect in the duodenum is neutralizing the gastric acid, which raises the pH, because alkaline means more pH, which is optimal for the pancreatic enzymes because the pancreatic enzymes include the lipase, amylase, and proteases. Secretin increases the bicarbonate by stimulating pancreatic and biliary secretions, right? So also something, yeah, it trapped in these Brunner's glands, the bicarbonate is trapped in mucus that covers the gastric epithelium. Okay, we stop here.